uh, relevant when we're talking about microscopy, right? So you got a cell and you're looking at it under a microscope. So there is the actual size of the object and there is the image size that you have, right? For example, agar there's a cell on a slide within a microscope placed on the stage, niche se light source hoga, right? And you're getting an image to your eyes through the lenses. So what you're seeing is an image, and what is placed on the slide is the actual cell. Yeah.
Yes.
Right. So what you see is a magnified image, a much larger image than the actual cell, right?
Yes.
So therefore, if you take the image size and divide it by the actual size, you get what is the magnification. Okay. Now, 1 centimeter is equal to 10 millimeters. 1 millimeter is equal to 1000 micrometers. 1 micrometer is equal to zero point 0.1 millimeter, 0.001 millimeter, or 1 into 10 to the power minus 3 millimeter. Yeah. Right.
Yes.
So magnification does not have any units. So it's 50 times bigger, 5,000 times bigger, right? But it does not have any units. Can you tell me why that is?
Not sure.
Not sure. Okay. So when we're talking about the image will have a size, right? So the image will have a length XYZ, let's say 1 centimeter, right? Which is 10 millimeters. And the actual size would be somewhere around 2 micrometers, let's say. Right? Now what's 2 micrometers in millimeters? It's 2 into 10 to the power minus 3. So you divide these two. The two units will cancel because they're the same. And then you have this value. So this is, we'll take this upstairs and we'll make it 10 to the power 3. This becomes 10,000 over 2, which becomes 5,000. So that magnification is 5,000 times.
Okay.
Okay. So can you see that it has no units? No. Now we're moving on to a concept called movement in and out of the cell or transport in and out of the cell, whereby we will learn what are the modes of transportation in and out of the cell, right? Transport of substance in and out of the cell,
Yes.
right? For example, you will have different minerals will go in and out of the cell, different nutrients will go in and out of the cell, and they have different modes of transport. Some require energy, some don't require energy, some require a transport protein, a channel through which they can go. Some don't require a channel, some can directly move through the cell membrane. Yeah. So diffusion. Diffusion is the net movement. Net movement means overall movement of particles down the concentration gradient, high to low. For example, if I have A and B. A has a concentration of 5 moles per milliliter of sodium, and B has a concentration of 2 moles or 0.2 moles per milliliter. Where do you think the particles would move?
To B. To A. Sorry.
From A to B. Okay, from high to low. So this is a higher concentration and this is a lower concentration, right? So this moves from A to B. The particles of sodium would move from A to B, and that would happen up until both of these concentrations are equal. For example, if you take the average of these two, it would come out to be 2.7, 2.6, right? So both places would become 2.6 moles concentration, right? Whereby the movement would stop. If the movement is stopping, though the two uh, solutions will be called isotonic. And this place where they will be is called equilibrium. They will come to an equilibrium. There will be no movement in the substance anymore, in the solvent, right? So we'll come to these type of solutions afterwards. We'll have hypo, hyper, and isotonic. So diffusion is the net movement of particles from high to low due to random movement. Yeah.
Yes.
Okay. Energy for diffusion comes from kinetic energy of the random movement of molecules and ions. If you studied in chemistry, particles have their own energy. That's called potential energy, but that is translated into kinetic energy, which causes vibration, right? Or movement, which is their own movement. It is built into them, right? So they have their own energy, they have their own movement, right? Each particle. As a result of this motion, we have diffusion. Okay.
Yes.
The diffusion of gases and solutes is important as without it, molecules that are needed for life, for example, glucose and oxygen for respiration, would not be able to get to the places they are required to get. Some substances move into and out of cells by diffusion through the cell membrane. Okay. Factors that influence diffusion. Okay. So this is an important part. This is something that they will ask you often. So concentration gradient. So how high to how low? Yeah. Temperature. Temperature causes faster diffusion because temperature increases the movement of particles. Yeah. If at any point you
Yes.
don't understand what I'm saying, stop me there.
Okay.
Surface area to volume ratio. When you have an increased surface area, right? For example, you have an increased surface area. uh, right? You have increased surface area, or you have a smaller surface area. This will have a greater rate of diffusion than this. Even though
I
the
don't
volume of these
understand.
even though the volume of these two might be the same, if there is an increased surface area, there is more possibility of diffusion, right? Compared to just this.
Oh, okay.
Okay. Distance. The distance between the particles and the place where they need to get, right? Because energy is required. Okay. And if the energy is there, but the distance to cover is a lot, they would lose their energy and won't be able to diffuse. Okay. Now, what's osmosis? Osmosis is pretty much the same as diffusion. However, it is regarding water molecules.
Okay.
Okay. The cell membrane is partially permeable. When we're talking about something permeating, it means it is moving through something. Okay. So you're having movement through an object, right? So you're permeating it. It's saying partially permeable. The cell membrane is partially permeable. So if there is one thing, it might let it go. And if there is another thing, it might not. You understand? So it's selectively permeable or partially permeable. So it is selectively permeable or partially permeable. Therefore, it can let some substances through without hindrance and some substance it will not let through. Okay, understand?
Okay.
For example, it's a gate with an ID card checker, right? So if you have the ID, you can go inside. If you don't, you can't. Okay, then you need a special pathway. Yeah.
Yes.
Okay. So small molecules like water, they can go through directly, but larger molecules cannot. For example, glucose is a much larger molecule than water, right? Glucose is C6H12O6, while H2O is just H2O, right? So you can compare the size difference. So osmosis is the net movement of water molecules from a region of higher water potential to a region of lower water potential, right? Through a partially permeable membrane. Okay. Which bit of this did you not understand? So we'll focus on that.
I've understood it so far.
Okay. So when we're talking about water or osmosis, we'll talk about water potential.
Yes.
Okay. And if the solution has a higher water potential, means it has more water molecules. So it is a dilute solution. Okay. Are we good till here?
Okay. Yeah.
Right? If it has a lower water potential and the water molecules are less, so that means the solute is more. Therefore, it's a concentrated solution. Yeah.
Yes.
So concentration of solute in cell low in so okay. So now there are three conditions, right? Under which a cell can change its morphology or the shape, basically, right? If you put it in a solution which has low concentration, which is basically a concentrated solution. A concentrated solution, right? So low water potential, high concentration. Yeah, the cell will shrink. Why? Because water will leave the cell.
Oh, okay.
Okay, so it becomes flaccid,
Okay.
and the solution that it's put in is hypertonic. Hypertonic. Tonic is referring to the concentration of solute. So hyper means more solute, right? If it has more solute, it has less water potential. Less water potential, so the water will be sucked out of the cell because it has a higher water potential. And the cell becomes flaccid. Isotonic is a solution which has the same concentration of solute, therefore, it has no change on the cell. However, if the cell has more concentration of solute and if the cell has more concentration of solute, right? So lower water potential, and outside you have a dilute solution with high water potential. The water goes into the cell and makes it turgid or best. We'll come back to this in a second. And the solution is called hypotonic. Explain why that is.
It's basically the opposite of hypertonic.
Yeah, hyper means a lot of solute, and hypo would mean less solute. Yeah.
I'm a little confused on the solute part.
Okay, we're talking. This is talking about the solution that the cell has put in. Okay,
Okay.
right. यहाँ पे we said that it's a dilute solution, right? So the concentration of the solute is low. So it's a hypotonic solution. Here it's a high concentration solution. So it's a hypertonic solution. Okay,
Okay.
right? So let's keep it this way. This is our cell. Okay. Now, if the concentration is high in here and the concentration of solute is low in here, so when we're talking about water potential, let me just change the color. The water potential would be high outside and low inside, right?
Yeah.
Right. So water moves in. Yeah.
Mm-hmm.
Okay. Now, what is the solution outside though?
Low.
Low, right? So the concentration is low. So this is hypotonic, right? Now, considering the same thing, you have higher concentration inside, lower concentration outside. Water potential inside is lower, and water potential outside is higher. I think I drew the same thing again. So, if you have a lower concentration inside, higher concentration outside, water potential lower outside, water potential higher inside. So, water leaves the cell.
This is hypertonic.
This is hypertonic. Yes. Okay. Okay.
Yes.
Now, we talked about the cell bursting or lysis. This is the concept we have in animal cells. Now, animal cells don't have a structure which is present in plant cells. Do you remember what structure that is?
The cell wall.
Cell wall, yeah. So the cell wall protects plants from bursting, and it causes a phenomenon called turgidity. It makes the cell very, very strong, right? So, for
Mm-hmm.
example, if you have a very strong outer covering and you fill that with a substance, it would become tough, right?
Yes.
Right. So that's basically what happens in plant cells. However, plant cells have a cell wall. Animal cells do not have a cell wall, right? So they don't have that extra protection. What happens is when they're filled up, they explode, they burst, they cause lysis, cell lysis, which is called the bursting of a cell.
Okay.
So this is the cell becoming flaccid, this is the cell becoming turgid, this is the cell becoming plasmolysed in a plant cell having lost water in a highly hypertonic solution. Okay. Now there is something called a dialysis tubing experiment. So what is a dialysis tube? Do you know what that is? Have you heard the term dialysis?
Probably.
So people, when they have dysfunctional kidneys, they undergo dialysis. Okay. So
Okay.
what it does is, what do your kidneys do? Do you have a basic idea of what your kidneys do?
I guess, yeah.
So what they do is they filter out waste material and they take up the important stuff back, right? So they
Yes.
are in a sense also partially permeable. They're not partially permeable, they have their own functions, right? So they can take stuff up, they can release stuff out. So the dialysis tubing, it mimics the kidney. It mimics the kidney in the sense of filtering out the waste stuff and putting back the important stuff. For example, glucose is taken back up, some sodium is taken back up, some potassium is taken back up, right? The rest is waste material. You've got a lot of urea that's taken out. Okay. So this tubing is used in order to see the movement of substances in and out of the cell in order to mimic it. So it's made from cellulose. What else is made from cellulose? Do you remember what else is made from cellulose?
No, I don't think so.
A plant cell wall, right? Plant cell wall is made from cellulose.
Okay, okay, okay.
So pores are small enough to prevent the passage of large molecules, for example sucrose, but allow smaller molecules such as glucose and water to pass through diffusion and osmosis. Through these processes, we have the removal of water and glucose, right? And but sucrose is left behind. So this is your sucrose solution. This is water outside, and this is your whisking tubing, which is partially permeable. So what you'll see is the level of water will increase or decrease. उसके हिसाब से, right? So water moves
Yes.
into the whisking tubing, which will make the level of this rise. Active transport is the last thing that will do. Active transport is when you have. Acha, ab tak whatever we discussed, it wasn't using energy, right? It was going as a natural phenomenon. It was happening on its own. It did not require energy. It was passive. Passive, which is without energy,
Yes.
right? Now, active transport, the first thing, the first word suggests that energy is used.
Could you repeat that?
Yeah?
Could you repeat that? I couldn't hear you.
Active transport, the first word suggests that energy is used because it's active, right?
Yes.
So, the movement of particles through the cell membrane from a lower concentration to a higher concentration against the concentration gradient and uses energy from respiration. So, up tak whatever we saw, it was going from high to low. High to low is the natural phenomenon. It's how diffusion occurs. It's how osmosis occurs. Yeah. But when we're talking about active transport, it requires energy. But why would it need energy? It needs energy because it's going against the natural phenomenon, which is diffusion or osmosis, right? If we're talking about particles, it's diffusion. If we're talking about solvents, it's osmosis, right? But now we're moving against the concentration gradient, going against the natural phenomenon. So energy is required. Yeah. An easy way to visualize this is if you have a ramp, right? And you have a boulder. Let's put a ball here, right? So if I push this slightly, I might not even need to push it. It would just move downwards due to gravity, right? Yeah.
Yes.
from high to low. But if it is placed all the way down here and I want to bring it up here, would I require some energy because it won't go up there on its own, would it?
No, I won't.
Right? So this is active transport. So I am giving it energy and I'm bringing it all the way up here through energy. This is active transport. Okay.
Okay.
Carrier proteins or protein carriers are also used during active transport. So this is what we're talking about initially when we were talking the special entry doors. Okay. So they're embedded into the cell membrane like gates, and it picks up specific molecules and moves them through the membrane against their concentration gradient. So this is the persuasion required, right? So it won't just need energy, right? It would need a person pushing it as well. So this is the person who is pushing it, right? And this is the person who is expending this energy in order to push it upstairs, right? So this is that person, and energy is required to make this protein work. Yeah, understand?
Yes.
Okay. So the active transport is needed when an organism wants to optimize the nutrients it can take up by the ion uptake. For example, ion uptake by the root hair cells. Right. So ions like magnesium, some other minerals as well, they're present in the soil. Yeah. And they're already in a high concentration within the root hair cell. However, they're still required in a larger amount by the plant. So the root hair cells will expend energy in order to take them up anyway. Okay. So this is
Yes.
an example of active transport. Another reason active transport is used is some particles are too large, right? They have a larger size to cross the membrane by diffusion or active transport. So some specialized cells have developed method of taking up these particles, and they are engulfed by the cell surface membrane flowing around them. This process is called phagocytosis and is used by white blood cells. So, giving a basic overview of this, you have immune cells. It's immune cells, right? Which are WBCs, basically, right? What these WBCs do is they'll find some sort of bacteria, right? And this is much too large in order to be taken up by the cell on its own, right? So we have this cell here, and what this cell will do is it will come close and it will change its structure, right? And it will engulf this cell or this bacteria, whatever this is, right? And it will take it into itself, right? And this process is called phagocytosis.
Oh, okay.
Okay. Phago means eating. Okay. So this is called
Yes.
phagocytosis because apparently the white blood cell eats the bacteria.
That makes sense.
Okay. Another example of the same thing but slightly different is if there is a liquid material, right?
Yes.
And the particle takes it up, the white blood cell takes it up. It's called pinocytosis, which means drinking. I don't know if that's in your syllabus, but a bit of extra knowledge, right? So
Okay.
that's all about transport within the cell, out of the cell. Any questions?
No.
Okay. This another name for this is also called bulk transport because a lot of material is taken up in one fell swoop. Right? So it's also called bulk transport.
Okay.
Okay. Any questions?
No.
Okay. I'll upload the lecture as soon as I get time.
Okay. Thank you.
Okay. Lapis.
Allah Hafiz.